Today we will be discussing HLA and how it relates to blood banking. So HLA is very complicated. It is the most polymorphic system, so it intimidates a lot of people. Blood bank does have some exposure to HLA due to platelet refractory patients and a few other things we'll discuss in this lecture. But unless you work in an HLA lab and you're exposed to it in a great capacity, it can be very confusing. So the HLA system has a large amount of functions. They help recognize the self from the non-self. They help antigen present and the immune response. And they coordinate the cellular and humoral immunity. In practice, HLA is important in transplantation, platelet transfusions, febrile non-hemolytic transfusion reactions, transfusion-related lung injury, post-transfusion graft versus host disease, and they have some disease associations. So even HLA nomenclature is very confusing. When we write it, it's like this. It starts with HLA, and then it states the gene, so it could be A, B, DR, DQ, DR beta is what DRB stands for. It has the asterisk as a separator. Field 1 is the serological antigen or allele group. Field 2 is the allele variants in that group. And then it goes further for all the nucleotide substitutions. If there's a letter at the very end, a suffix will denote changes in expression. So it could be low, secreted, it's present in the cytoplasm, or N for null, which are alleles that are not expressed. The major histocompatibility complex is a large gene family associated with intercellular recognition of self and non-self discrimination. In humans, the MHC is called HLA, human leukocyte antigens. The genes are on the short arm of chromosome 6. You can see they're all very close together, which explains why they are inherited as a haplotype. And this is just another view on the chromosome and then blown up where all the genes are located. So the MHC genes are autosomal and they are codominant. So you will inherit one haplotype from your mom, one haplotype from your dad, and they will both be expressed. So expression of HLA antigens can vary. Class 1 can be downregulated in malignant conditions like bone marrow cancers, viral infections, certain stress conditions can downregulate. Class 1 and 2 can be upregulated by cytokines, tumor necrosis factor, and interferon. And HLA-C is expressed very low on platelets. That's why when we're doing platelet testing, we only test for A and B loci. HLA class 1 molecules consist of two protein chains, a glycoprotein heavy chain encoded on the short arm of chromosome 6, and a light chain called beta-2 macroglobulin encoded by a gene on chromosome 15. The heavy chain penetrates the cell membrane, but the beta-2 macroglobulin does not. Beta-2 macroglobulin associates with the heavy chain through the non-variable alpha-3 domain. The external portion of the heavy chain consists of 3 amino acid domains, alpha-1, alpha-2, and alpha-3. Class 2 molecules consist of two structurally similar glycoprotein chains, alpha and beta, and both penetrate the cell membrane. Each chain outside the membrane has 2 amino acid domains. The outermost domains contain the variable regions of the class 2 alleles. The outer domains of class 1 and 2 molecules fold to form a cleft, or a peptide binding groove. Exons code for variable amino acids forming unique binding grooves which allow each to bind peptides of different sequences. This helps to explain the reason HLA genes are the most polymorphic of the human genome. Presentation of MHC-bound peptides to CD8 and CD4 T cells promote immune regulation through self and non-self recognition. On the other hand, enormous allelic variability of HLA creates a barrier to transplantation. HLA class I antigens are found on all nucleated body cells. HLA class II are found on macrophages, dendritic cells, and B cells, which are antigen-presenting cells. Most antigens are proteins that are presented to immune cells by antigen-presenting cells as small peptides bound to either class I or class II molecules. These antigens require T cell help to elicit an effective immune response. Once transported to the cell surface, class I molecules interact with CD8 positive T cells. Class II molecules interact with CD4 positive T cells. These are responsible for the production of antibodies. Since HLA antigens are highly immunogenic, they also are relevant in complications of transfusion therapy, such as platelet refractoriness, febrile non-hemolytic transfusion reactions, transfusion-related acute lung injury, and transfusion-associated graft versus host disease. In pre-transfusion testing, there can be HLA remnants left on red cells. These are called Bennett-Godspeed antigens, BGS. An anti-BG in the patient's plasma can cause confusing reactions in the antibody screen and panel testing. Reactivity is generally weak in the IAT phase, and these antibodies are considered clinically insignificant. So mostly they just cause confusion during these workups. One of the main reasons we interact with HLA testing in the blood bank is platelet refractoriness. This can be caused by non-immune or immune-related causes. Febrile non-hemolytic transfusion reactions may be induced by HLA, granulocyte, platelet-specific antibodies, or biologic response modifiers such as cytokines, complement fragments, or lipid compounds found in the plasma of stored platelets. Pre-storage leukoreduction by filtration or apheresis collection has reduced the incidence of this phenomenon. TRALI, or transfusion-related acute lung injury, is a complicated event that is believed to be induced when donor HLA antibodies activate neutrophils in the recipient's lungs, which cause an increase in cytokines, which cause very acute lung damage. This is why we test all plasma products for HLA antibodies. AABB, who publishes standards for blood banks and transfusion services, released a new standard in 2014 stating that all women with previous pregnancies must be tested for HLA antibodies. And any units that are positive for HLA antibodies are not suitable for allogeneic transfusion. Transfusion-associated graft versus host disease occurs when there's a 1 HLA haplotype match between the donor and the recipient. So the donor T lymphocytes attack the foreign recipient tissues. The recipient does not attack back. This is usually in immunocompromised patients. This is why when units with common HLA types from family members must be irradiated prior to transfusion. And then we see graft versus host disease in transplantation. So it can have a very diverse set of symptoms. So this is the mode of action in donor-specific antibody-mediated graft damage. The complement cascade is activated. There's interaction with HLA and non-HLA antigens on the cell surface. and then antibody-dependent cell-mediated cytotoxicity takes place. There is also some relationship between disease associations and HLA typing. Here's a little list. Basically, if you have this HLA type, you may be more at risk for having these diseases. So now we'll talk about the methodologies for HLA typing. Originally, HLA typing was done by serological methods. This is no longer acceptable, so it's not specific enough. But it was basically done in a CDC, a lymphocytotoxicity assay. Now it's only performed by molecular typing methods. So we have PCR-based assays. SSO and SSP are the main methods. SSP is sequence-specific primer, direct amplification method. Those can be visualized in gel or by real-time PCR. And then we have SSO, which is sequence-specific oligonucleotide, which is a probe hybridization method, which can be visualized by flow cytometry, such as in Luminex testing. And then there's sequence-based assays, which you can use Sanger sequencing or next-gen sequencing. So the cytotoxicity assay that was performed, you basically had antibodies for known antigens plated out. You had to isolate the patient's lymphocytes and add them to this well, and then see which ones were killed by the antibodies. So if you had a anti-B15 in one well, and then you added the lymphocytes, and then it was killed, then you have a positive reaction, and you're assuming that that patient is B15 positive. The SSO method, you can see we analyze with the Luminex assay. We isolate the DNA, do a PCR amplification of the HLA sequences, and bind it with the bead that has a known sequence, and add a label so that we can run it through the flow, in this case Luminex, and then the analysis software will give us an HLA assignment. So in SSP, we're targeting a sequence-specific primer. So if we're looking at this area as something that's going to determine if something is a B27, then we're going to make primers that will amplify this area, and then we will run it on a gel, and then we'll see the amplification. Usually you'll have to have many wells because the HLA is so polymorphic. Certain antigens will have common alleles, so you'll look at the combination. Maybe B27 is positive for these three, and then B59 is positive for these three. So you'll have a bunch of different sequences, gel it, and then look at what all the positive wells tell you. Sanger sequencing is a method of DNA sequencing that involves electrophoresis and is based on the random incorporation of chain-terminating dideoxynucleotides by DNA polymerase during in vitro DNA replication. It was first developed by Frederick Sanger, and it is the most widely used sequencing method for about 40 years, which is now being kind of taken over by next-gen sequencing, but it is very useful. Next-gen sequencing is quite complicated, and it varies based on what platform you're using. So Illumina and PacBio have completely different methodologies. There is a link right here if you want to watch a little video about how Illumina sequencing works. So we talked about HLA antigen typing. Now we're going to talk about HLA antibodies detection. You can use CDC, although it's not really used anymore. Usually you're using flow cytometry or solid phase detection. So in solid phase detection, which an example would be the Luminex bead technology, you have these Luminex beads that have HLA antigens bound to them. You're going to incubate it with your sample, serum, or plasma, which may or may not contain HLA antibodies. If the HLA antibodies are present and they bind to the HLA antibody, when you add an anti-IgG, it will fluoresce. And when you run it through the flow, you'll get raw data, which is expressed in MFI, which is mean fluorescent intensity. Flow cytometry methods are very similar. You have beads that have HLA antigens bound to them. You incubate it with your sample plasma or serum, and then you add an anti-IgG that has a fluorescent stain bound to it and run it through the flow. If you have fluorescence, it will look similar to this. Your negative control will be a low fluorescing peak, and then if it is positive, you'll have a bunch of peaks depending on the specificity of the antibody. If you have one specificity, it may look like this, and then if you have many different specificities, you'll see lots of peaks like this. So cross-matching in HLA is much more time-consuming than cross-matching in blood banking. Basically, you need to isolate the donor lymphocytes, which you may get peripheral blood that you can isolate from, or if it's a deceased donor, you'll get lymph nodes, which you can see here. So you're basically going to get all the lymphocytes out, isolate just the lymphocytes, and lots of times it looks very messy like this. And then you're going to incubate the lymphocytes with patient serum to see if there's a reaction. So the other cross-match that we perform is the CDC cross-match. We isolate B cells and with the patient serum, and then we add complement and IgG and see if there's death. So when you look at this under a fluorescent microscope, which you'll use a stain, usually we use acridine orange. If you see dead cells, it'll look like this, and then alive cells will look like this. ASHI has a specific scoring system for the CDC crossmatch, so you can see that down here. If there's 10% or less dead, you call that a 1. If there's greater than 81% dead, you call that an 8. And then the flow crossmatch, like we discussed before, you're incubating the patient plasma or serum with the donor lymphocytes, and then you're adding a fluorescently labeled anti-IgG, and what you'll see is similar to this. If it's negative, you'll have very low fluorescence. If it's positive, you'll have more fluorescence like this. Engraftment monitoring is something else we do in the HLA lab. We use STRs, short tandem repeats. This is also what they use in forensics to determine who the culprit could be, or in parentage testing to see who the father could be. Basically, each human has a different STR profile, so you can compare that to other profiles and determine the likelihood that that DNA came from a specific source. So we use this to monitor patients after bone marrow or stem cell transplants. We take their STR profile before transplant and the donor's profile before transplant, and then starting at 30 days post-transplant, we take a sample from the recipient and see if they have transitioned over to the donor profile. So each one of these is a different loci. So for example, for TH01, the patient was 6 and 9.3, and the donor is 8 and 9. And then post-transplant, you can see the patient has a little of everything. So we do a calculation and see that they are 38% donor. So we want to see them transition 100% to donor after transplant. And that's a good segue into the NMDP, the National Marrow Donor Program. Anyone can sign up for this. They'll send you a swab in the mail, and then they'll do your HLA type. And your DNA type will go into a registry so that if a patient needs your HLA type, you'll get a call and ask if you're willing to be a donor. You do not have to donate actual bone marrow anymore. They can give you a drug that will induce stem cells to be released into the peripheral blood. So you can sit in a machine and get those cells by apheresis, just like you're donating platelets, and you can save a life. If you're interested, you can check out this website, sign up, and they'll send you a little kit in the mail. I'll also give a little plug for organ donation. I did work in an HLA lab for a while, and it is pretty insane the amount of people that are on organ wait lists. So you can look at the UNOS wait list to see how many people are waiting for organs, and it is pretty insane. So I always recommend, if you don't have a strong feeling about it, why not donate your organs? And also, some people are not aware that you can donate your cord blood. This is something that normally goes into the trash, but it is full of stem cells. So if you are pregnant or going to be pregnant or know anyone that's pregnant, you can let your doctor know that you're interested in cord blood donation. And I have some websites here if you want to check out more information about this. It just looks like a small unit of blood that they collect from your cord blood after your baby is delivered. And then we isolate the stem cells, we freeze them, we also do the HLA typing, and they go onto a registry. And usually these are used for, because it's such a small amount, they're used for babies or children. So whenever a child needs that HLA type, we pull that frozen cord blood out of the liquid nitrogen, and it's transfused as a stem cell transplant, and it can save that baby's life. So it's either going in the trash or saving a life. So you can look at it that way. But I highly recommend doing this. And that was all. Keep in mind, I know HLA is overwhelming. You don't need to know all this information. It's just kind of just an introduction to give you kind of a feeler about how complicated HLA is and how it might work into the blood banking world.